to our inaugural session of the Casey and I speaker series. I hope that you'll all join us for continuing with our speakers throughout this series. I'm going to just share my screen and start the presentation. And so what I wanted to do today was really give a bit of a perspective from the center, from the Krembil Center for Neuroinformatics, on how we're approaching an integrative multiscale perspective on brain health. And to do that, I'll take you through first an introduction to the center. So first of all, the Krembil Center for Neuroinformatics, we inaugurated officially about 1 year ago. So we're coming up to our 1 year anniversary, and we'll have a nice day of events and presentations in greater depth than this one. And at the core, one of our fundamental challenges is how do we capture the right kind of data from the clinic that's relevant to really inform treatment, classification, diagnosis, stratification of patient populations, and then ultimately start to identify the biological underpinnings of mental health disorders. And so this core challenge of multi-scale data, integrating high-quality data, including genetics, imaging, and mobile data, is really one of our pillars. Now, once you have high-quality data that you trust, that you can rely on, there's a lot of opportunity with machine learning tools to make diagnoses better from the data, predicting mental health disorders, outcome trajectories, and so on, using modern machine learning techniques. So artificial intelligence is another pillar for this. Multi-scale brain modeling is something that I'll take you through today as well, which is really using computational modeling. A lot of it is actually using machine learning as well, but at different levels of organization of the brain. So looking at how can we identify, from genomics to cellular, to synaptic, to circuit, to really build a deeper understanding of mental health disorders. Now, all of this is done at the center in an open science, collaborative team environment. And a core part of our mission and mandate is to train the next generation of scientists. So we have a lot of educational activities, workshop series, and courses, including a Casey and I summer school that I encourage you to look up. In addition, we have a mandate to really look at how do we accelerate the translation of technologies, algorithms, and so on to the clinic through an innovation initiative. The way that we approach this challenge of kind of the research world and the clinical world, and how do we really operate between them, is truly collaborative. It requires close collaborative partnerships in the clinic, requires collaborations, of course, with the researchers, but also the technology developers, the informatics infrastructure to actually get the data out and integrate it into a form that we can search it, we can query it, we can analyze it, and then use that to build computational models, both machine learning and also simulations of brain circuitry. And this is not the sort of thing that happens once, where you make a waterfall plan and this is a project that we're going to achieve once and we're done. It's much more about establishing an iterative, collaborative, and integrative project and series of projects that form collaborations between the clinic and the research, and so much so that the lines start to blur. And that's because we really want to bring a multi-scale perspective on these disorders, understanding the underlying biological mechanisms to bear, to be relevant in the clinic, and also towards developing treatments and therapies. So here you can see, one of our challenges is that the current approach to diagnosis is based on a symptomology rather than the underlying biology. And that's one of the grand challenges, is how do we get towards a definition of mental health disorders based on the underlying biology? And that requires looking across the scales from the genomics all the way up to the individual and populations in order to better understand what really defines a mental health disorder. Now, we've got a phenomenal team here at the center, and really each team is tackling a different level of organization in the brain, and then they all work collaboratively, both within the center across teams, but also externally, as you'll learn about, with other research teams in order to start to build this integrated perspective. And so the first team is led by Srijoy Tripathi in computational genomics. Itai Hai leads the team on brain circuit modeling. John Griffiths leads the whole brain modeling team. Andrea Diaconescu leads the cognitive network modeling team. And Leon French leads the integrative neuroanatomy. And Daniel Felsky leads what we call the whole person modeling team. And I'll talk more about their work later. We also have a flagship project called the Brain Health Databank. This is led by Dr. Joanna Yu. She's the senior project manager. And in collaboration with Stefan Kloiber and Jeff Daskalakis, this is really starting with major depressive disorder clinic, the ICP, the so-called integrated care pathway, to see how do we establish a process and the infrastructure that can accumulate high-quality longitudinal data about individual patient-level journeys and trajectories of their treatment in order to better understand, build a better integral picture of each individual, in order to make predictions and models that can help. So part of that is deploying tablets and tools to the clinic to get the engagement and the buy-in in the collection of the data, and making it valuable to day-to-day clinical operations to have that data visualized as a decision support tool. And then we're also building the infrastructure to layer in research studies integrated with the care pathways so that we can, for example, very often clinicians will want to know, how did my patient sleep in the previous week? And we can do things like layer in a wrist-worn actigraphy device that can give a measure of sleep for each individual, and we can integrate that into the decision support tool. All of this is really with the aim to accumulate enough high-quality evidence across different levels and across disorders that can help us have a data-driven approach to defining and treating mental health disorders. And this is an example of a decision support tool that's been developed, co-designed with the clinic to show an individual patient's trajectory over time, over multiple visits, to show their PHQ-9 measure, their risk scores and medication and weight, and so on, so that at a glance, a clinician can get a view on each individual patient's history and trajectory, and use that to better inform the treatment. So as we start to think about integrating kind of the neuroscience and the biological mechanisms into a care pathway, we've got some major challenges, because there's a big gap between being able to measure the underlying biology in the clinic and where we are today. So one of the starting points is just an observation that virtually all neurologic, psychiatric, and substance abuse disorders are associated with changes in excitability in different parts of the brain circuits. And this is a remarkably consistent finding. And so there's clearly some important principles to be learned and to bring to bear on this understanding. And one of the approaches that is important to understanding what role excitability in neural circuits can have is to use integrative multiscale models of brain circuitry. And I'll take you through an example of one that was built, as David mentioned. I was previously at the Blue Brain Project, and this is just a quick example of kind of reconstructing a brain circuit in order to understand some principles of excitability and changes in excitability and what the impact is. So the way that this process starts is really to get a list of the building blocks of a brain circuit. This was built, the blue brain circuit was built from data collected over many years about one single brain circuit in a rat. This is characterizing individual neurons, all of the different types of neurons, their morphology, so their three-dimensional shapes, their electrical properties, their synaptic properties, how do they communicate with each other, their composition in terms of, which ion channels, which are these membrane situated proteins that govern their excitability, what's their composition, how many cells are there, how are they connected, all of this reverse engineering of one brain circuit, which is about 0.5 millimeter wide and 1.5 millimeters tall. So capturing all of that data and then using it to actually reconstruct digitally in a computer a version of that circuit. And so this is some of those building blocks. These are the diversity of cortical neuron types. And one of the things that we've learned is how important this diversity of cell types are in actually building a robust cortical circuit. You can see that each class of neurons has a different shape. There's inhibitory neurons, there's excitatory neurons. They've got characteristic shapes, just as certain different species of trees have characteristic shapes, but every single neuron is unique. And we use the statistics and the data about the composition of that circuit to start to build a digital reconstruction by placing these neurons in a three-dimensional volume comparable to what exists in the biology and start to build a virtual reconstruction. And so this is, in this case, 31,000 model neurons positioned according to their statistical densities and distributions. And what you can see is, as we zoom into the center of this while it's being built, is the placement of the neurons is actually creating a virtual circuit because the axons are coming close to the dendrites, and that's forming the foundation of the connectivity. When we layer in many different rules governing the connectivity based on a lot of biological detail, axon, bouton densities, the numbers of synapses per connection. We do a lot of validation to make sure that this is capturing as accurately as possible the statistical properties of this circuit. And when we put all of that together, again, in a purely data-driven way, this circuit gives rise to emergent oscillations and activity. And this activity, so this is the color code is showing the voltages. Dark blue is when it's hyperpolarized and quiet. When it gets excited, it gets brighter red and flashes white when it spikes. And all of these neurons are then interacting and collectively generating this slow wave activity, this slow oscillation that is really an intrinsic characteristic of cortical circuitry to generate this rhythm. And so that emerged spontaneously from the computer simulations without us tuning for it specifically. And then we could use that to build a virtual brain slice and do additional experiments. And this is important because the validation requires many, many additional virtual experiments to make sure at different levels that the circuit corresponds to a biological entity. And one of the key things that we found here is that when you've got increased excitability, and this is getting back to that point that in many different brain disorders, there's a disruption in the excitability of different circuits. And in this case, in cortex, if you have a hyperexcitable local circuit, you can see on this top bar of the bottom section, the hyperexcitable condition creates this all or nothing response where once you hit the threshold of input, you just get this broad traveling wave, a very strong response, which is not very specific. Whereas if you have normal excitability, there's really a direct proportional response to the input where the characteristics of the input are preserved. So your signal to noise ratio is much higher in the bottom trace than in the hyperexcitable. So the normal excitability has a higher signal to noise ratio. So this is a general property of neural circuits, particularly in this case in cortex. And we want to explore that further in order to understand specific mechanisms underlying brain disorders. So in a collaboration, well, so separately actually, Professor Etienne Sibyl has been studying for a great amount of time core cortical microcircuit in the cortex, and looking at the role of somatostatin positive neurons. So this is one of those types of neurons that I showed you earlier, one of those building blocks. And what I'm showing you in the middle here is how that neuron, a somatostatin positive neuron, which is an inhibitory cell, innervates pyramidal cells, which are excitatory cells. And this is a very core circuit of cortex. And it turns out that when you have a disruption in that inhibition, if you have a decrease in that inhibition, you actually create a disruption in this circuit with some deleterious effects. And it turns out that that reduction in depression occurs in many different disorders. So in Parkinson's, in Alzheimer's, in major depression, in schizophrenia, and in bipolar disorder, across all of these, there's a decrease in the SST GABAergic inhibition. And that's creating, in effect, a hyperexcitability. And that causes part of this observed change in excitability. And so this seems to be one mechanism of many potential, but one common mechanism across many disorders to change neural circuit excitability. And it's these very important somatostatin positive neurons. And so you can see there's quite a number of observations about this reduction in GABA across many disorders and in different neurons, and the effect of that on default mode networks. And there's a review here cited, so I really appreciate Etienne Sibyl providing these slides. Now, what's important is that when you reduce this depression, you're really impacting the excitatory signal to noise ratio. You're altering the information processing, and this is associated with cognitive dysfunction. And this very core circuit of the Martinelli cells, or somatostatin positive neurons, onto pyramidal cells really has a direct impact on cognitive function. And there's been a clear causal role for this reduced pathway in mood and cognitive symptoms, so across many different studies. I'm not going to spend a lot of time going through this, and Etienne will give a much more in-depth presentation on this at our open house. But what's important is that he's actually done a lot of work to develop and identify a compound that acts on this pathway. And that compound actually enhances then the inhibition that's been decreased. And by doing so, it reverses the cognitive deficits that have been seen due to that decrease in inhibition. It reverses some of the the cognitive deficits due to chronic mild stress in mice. It reverses neuronal morphology changes that occur due to this stress in hippocampus and prefrontal cortex. And it reverses cognitive deficits due to age. And so this is really a very remarkable work that by Professor Sibyl's lab, and it gives us a basis as a multi-scale computational team to look into, well, how do we understand better what exactly is happening within that circuit, within the changes in that excitability, and how can we better use that to diagnose and predict individual susceptibility and outcomes. And so, I just will conclude that section here, that this is a key pathway. Dr. Sibyl has a compound that acts on this, and augmenting that inhibition has potential for really improving cognitive and mood symptoms in depression and during aging, and really has neurotrophic and neurogenic effects as well in terms of restoring the health of the circuitry. And so coming back to the integrative multiscale modeling, how can we as a team and as the Krembil Center for Neuroinformatics collaborate with Dr. Seville to take his findings and build a deeper understanding and some opportunities for enhancing and collaborating with his team. And so Dr. Srijoy Tripathi, who leads the computational genomics team, is looking at how to use genomic sampling, both of the DNA and RNA, and to use known properties relating that also to, for example, the somatostatin cells, to infer the activity of those brain cells and the circuits, and to inform computational models to make a more personalized brain model that can help stratify patients in terms of their response to and the role of somatostatin cell inhibition. Dr. Itai Hai, actually, he was originally one of the members of the Blue Brain team and pioneered actually the model building approach. And now he's taken it to the next level, building computational models of both mouse and human cortical circuit activity. And in particular, he's looking with Dr. Seville at the impact of the somatostatin interneuron inhibition onto pyramidal cells, but really using computational models of those neurons, both from humans and from mice, and then testing in silico the pharmacology, the effect of a compound that would enhance that inhibition. And because he's computational modeling the full three-dimensional shape of these neurons, he can actually compute a simulated EEG or local field potential to start to understand the underlying synaptic and neuronal activity and how it relates to biomarkers that could be measured potentially in the clinic as EEG. And taking that further to the whole brain level is Dr. John Griffiths with whole brain modeling, looking at EEG biomarkers, again, building off of the work of the local microcircuit modeling from Itai Hai, and then bringing that to whole brain level models, both for rodent and human whole head EEG, and using that then to identify brain-wide dynamics and the impact of SST inhibition deficits on global signals and EEG distributed throughout the brain. At the cognitive network level, Andrea Diaconescu can use different cognitive tasks, in this case, potentially mismatch negativity, to look at essentially a surprise response within the brain, and then use that to subtype which networks are actually implicated in these alterations in excitability. And so by essentially inferring from the EEG network and from specific auditory or cognitive tasks to build models that predict which subtype an individual patient belongs to, and that can help enhance treatment and targeting of treatment for each individual, personalizing the treatment. Dr. Leon French leads the Integrative Neuroanatomy team, where he can use gene expression atlases to look for specific areas that may be more susceptible or more optimal for treatment with particular molecular targets related to, for example, GABAergic and somatostatin-related cell populations. And so that's another opportunity to align and integrate data in order to inform the relationship to somatostatin inhibition. And finally, Dr. Daniel Felsky leads the whole person modeling team, and there you can use co-expression networks of target genes and build up genetic scores combined with lifestyle and demographics in order to build better predictions of those whose brain activity may be impacted by vulnerabilities in the somatostatin pathway, for example. So this is again, looking for those opportunities to gather and integrate data and make predictions that can inform these specific pathways and inform better the stratification and prediction for each individual. Related to all of this is that virtually all neurologic, psychiatric, and substance abuse disorders are also associated with sleep disruption. So what does brain excitability and connectivity have to do with sleep disruptions? Well, it turns out, according to one of the leading hypotheses for sleep function, that this gradual increase in excitability and the increase of an excitability that we see, both during the day, but also, as part of a homeostatic process, but also in some of the excitability that we see due to possibly a deficit in inhibition can lead to increased slow wave activity. The slow wave activity is what you see in deep sleep, but interestingly, it's starting to be clear that happens other times as well. And that slow wave activity is part of the brain trying to restore synaptic balance, decreasing the synaptic density and strength in order to renormalize and rebalance the excitability of the local circuitry. And it turns out learning and other plasticity processes that increase the excitability actually create locally increased slow wave or sleep. So when you go to sleep after learning, and that learning involves one particular region, the slow wave activity will be higher in that region after a day's work learning that task. Similarly, if you suppress engagement or activity or plasticity within that brain area, you'll see a decrease in local slow wave. So part of what we're seeing is that slow wave activity and is actually not necessarily a global phenomenon of sleep, but can occur more locally concentrated during sleep. And it impacts the connectivity, the effective connectivity between brain areas. So testing using TMS from Marcello Massimini's group, they could see that when the brain was asleep or brain regions were in sleep-like states with slow wave activity, that interfered with the communication between brain areas, and computational modeling has been used to show exactly that effect as well. And what's striking about that is that slow wave activity actually can occur during wakefulness. And so you can actually have interruptions in connectivity between brain areas during wakefulness when you have this local slow wave activity. And this has been seen intracranially in humans after sleep deprivation. So if any of you have ever been sleep deprived, I suspect all of you have been at one point or another, and you feel like some part of your brain isn't quite awake. Well, in fact, it seems that there are local regions of your brain that can be producing this slow wave activity, which is effectively local sleep that is interfering with the communication between different areas of your brain, and therefore your cognition. And if this is sustained, if you've got chronic sleep disruption, or if this homeostatic process locally is disrupted for a long time, one of the things that you start to see is the hyperexcitability created by this local sleep deprivation can result in sustained microglia activation. And that's where they start to really, there's an inflammatory response, and they can start to eat a lot of synapses. And that may further impact and disrupt connectivity. And of course, it's part of an immune response and inflammation, which can lead to further neurodegeneration. So one of the interesting opportunities is to see how can we actually capture sleep and sleep disruption as part of the patient chart. And interestingly, using a wrist-worn movement assessment combined with the heart rate, you can actually get a fairly decent assessment of the overall sleep architecture of an individual. And that gives us a way to start to link brain excitability and the ability of the overall brain to engage in slow wave activity and relate that to behavior. and relate that to something measurable without having to go into the brain. So it's an opportunity. And that then can be used to enhance the overall, this multi-scale perspective can be brought to bear on that individual decision support tool for the clinician to provide a sleep analysis, to provide blood markers that are indicative of hyperexcitability or dysfunction in specific circuits, and using EEG and TMS EEG to assess locally the circuit excitability, as well as then potentially cognitive tests or auditory tests that can be used also to reveal brain network disruptions. So all of this is in the ambition and in the aim of really building up a richer, multi-scale approach to phenotyping brain disorders that bridges from the genetics, the cells and circuits, but also to behavior and to sleep and to cognition in order to better stratify and treat patients. I just give the credits here for the blue brain section that I showed you. There was 82 co-authors. And the team at the Krembil Center has just been a phenomenal team of informatics specialists. David Rotenberg is our operations director, and the whole team of scientists and IT team and our project managers and research coordinators. It's just a phenomenal team that keeps all of this running. And thank you very much for your attention. I hope I wasn't just talking to avoid that whole time and somebody heard something.
I think we heard you. Do you see the questions?
I do not. Let's see.
Okay.
Okay, very good. I see Peter, and now I do have the window open and open to questions. Thank you. Very, very good. Peter had no slow waves. Actually useful for restoring your plasticity, so your excitability. Any questions from anybody? If you type your question into the chat box in the Q&A section, does everybody have instructions on how to do that? Is that clear? Okay, just give it a few minutes. Okay, got a question from Hajr. How do you think the work of Casey and I will integrate with large imaging data sets like ABCD, for example? It's a great question, and I think there's a tremendous opportunity. One of the opportunities with large-scale imaging studies, also that are longitudinal like ABCD, is to look at changes in functional activity and effective connectivity, and also the networks engaged over time. And in particular, I think there's a lot of opportunity to look for signatures of sleep in fMRI signals, and potentially even seeing local disruptions in interactions due to local excitability or local sleep-like states. And I think the more that we can start to learn how to mine out of old signals signatures of the hyperexcitability that disrupts, of course, the whole default mode network, I think there's a great opportunity to start to better diagnose, and then, of course, understand the relationship to effective connectivity throughout the brain. And the work of, for example, John Griffiths, I think, in terms of building these whole brain models, will be very relevant. Other questions? So Andrea asks a question, is there any link between the therapeutic sleep deprivation and the GABAergic effects you discussed in the context of MDD? I think it's a great question, because we do see in some phenotypes of depression that sleep deprivation can at least have a transient improvement of symptoms for depression. And it's not in everybody, and it doesn't always last for that long. But I think that the question is is somehow is there an effect of the sleep deprivation causing maybe an increased recruitment of the inhibition of, for example, of these somatostatin neurons? And I think it's an open question there, and I think that it would be very, very interesting to explore the basis of that effect. So I don't have an answer to it, but I think it's quite interesting, and clearly, the opportunity of seeing how increased excitation, possibly through sleep deprivation, would impact the overall default mode network would be very, very interesting. And Dr. Kennedy asks, any thoughts regarding brain organoids as a modeling system? And I think it's a very interesting question. I think that in a way, part of what you get when you get an organoid is, of course, this collective set of course, the neurons and some glia. We're seeing that there are, at least in some of the organoid systems, you do get microglia, you do get glia, you do get neurons. Now, of course, the morphologies and therefore probably the connectivity of those neurons is not going to be the same as in a full-blown, geometrically full brain. But I think that there are physiological characteristics and possibly immune responses that could be looked at to see at the individual level. So if you're building a brain organoid from somebody's cells, that you may be able to understand some of the potential deficits or hyper responsive immune responses, possibly. I think at the physiological level, I think there's opportunity. I don't believe, could be wrong, but I don't believe that an organoid will really be useful for understanding kind of the wakefulness level activity and that potential for specific circuits being hyperexcitable, but I think it can be used for understanding the kind of core physiological, electrophysiological responses at a gross level. Let's see. Hud asks another question. Do you think we will get to a level where we can use machine learning to accurately diagnose patients, or is that the closest we will get as a second objective opinion in addition to the clinician? So that's a great question, of course, a bit controversial, and I do hope that we are able to accumulate enough evidence that the data becomes the primary driver for a diagnosis. It's going to take us a long time to get there, I think, because we've got a lot of work to do to get there, and to be able to validate and to be able to know that the data we're collecting is reliable and is giving an accurate kind of phenotyping of the individual. But I think eventually we will get there. Let's see. John Griffiths asks, what is the typical duration of local sleep intrusions? Are they on the scale of milliseconds, seconds, or minutes? Yeah, so a great question. I don't think it's really been robustly studied to know, what is typical. But I think on the whole, in the studies that I'm aware of, it's over multiple seconds where there's multiple waves, slow waves that are recurring. But of course, in sleep itself is a very dynamic process, and it's kind of going through phases of greater synchronization and less synchronization anyway. So I would typically say it's on the order of seconds. Okay, thank you, Kamal, for coming. And Andrea, let's see. In terms of the nature of collaborative work, does the center anticipate leading its own human subject research, such as imaging studies, or working collaboratively with other researchers across ChemAge and other institutions as partners on their research? So I think it's a great question. In general, we do see ourselves as a dry center, but collaborating with the clinic does give us an opportunity to actually lead human subject research as well. So as long as we have a clinical partner, and certainly there's a lot of opportunity there. We don't want to duplicate facilities. We don't want to duplicate efforts. We want to collaborate as much as possible. But I think that still leaves room to collaborate and lead some studies, for example, imaging or EEG studies, while collaborating with others. Joanna asks, is it possible to share questions with the group? That's being worked on. And then Yuchao Chen, are there any plans to further integrate and connect the KCNI team to preclinical researchers or the Brain Health Databank to preclinical data to facilitate, for example, experiments to verify model predictions? So yeah, I mean, I think part of what I showed you is actually at each level. So both, Sridhar Tripathi is working both with rodent, primate, and human data, and that's a very important kind of preclinical research to then connect to actual real human measurements. Similarly, Itai Hai is working both with mouse and human circuitry, and John Griffiths is working both with mouse and human. So, all the way up the line, we really see kind of this back and forth between understanding what can we measure and predict from rodent or primate, non-human primate, and bring that to bear on human studies, and to verify model predictions. And so that is very much an active part of what's happening. And of course, the collaboration with Etienne Sibyl is another branch of that, because he also has data from rodent experiments as well as humans. So that's absolutely a very important integration. And then Art Petronis asks, can you comment on what is happening with the US Brain Initiative? So actually, the US Brain Initiative has recently had a kind of a renewal for their next phase. So they're going to have another big investment, quite a bit of money, and they're going to start to move out of just their kind of non-human research. They're going to start to get into more human-oriented research. And of course, a big part of what they've been doing is building up a lot of data on individual building blocks of the brain and circuitry, understanding. So they have the Brain Initiative Cell Census Network, BICCN, and a number, I believe Sridhar Tripathi is also working a bit with them as well, but they're accumulating large amounts of single cell transcriptomic data, electrophysiological data, morphological data about all the cells or many of the cells of the brain. There's a part of that effort is reconstructing brain-wide neuron morphologies and starting to build, deconstruct, re-engineer circuits across the brain. And now, I think we'll start to see a lot of that being brought to bear on a deeper understanding that will start to bridge towards human studies rather than solely being focused on kind of some of the preclinical work.